Hello, everyone, and welcome to our webinar. My name is Amit Binderman. I'm the CEO of Cometa Bio. And in the next 20 minutes, we're going to talk about bone grafts, bone substitutes. We'll evaluate different types, and we'll talk about the differences of the properties between different graft materials and what we can expect. We're also going to talk about autologous grafts, which are very dear to me. And specifically, we'll talk about autologous dentin graft, which is what Cometa Bio is all about. Please save your questions to the end. Make a note of those questions so you don't forget. We'll have a Q&A session at the end of the presentation. Before we start, just a few words about Cometa Bio. We launched the company in 2014 with the aim to gain focus on autologous biomaterials, which is still considered to be the gold standard in order to improve clinical outcomes and increase predictability of bone augmentation procedures. Our product is obviously the Smart Dentin Grinder. Hopefully you're familiar with it. And this is a revolutionary device and protocol that converts extracted tooth or teeth from the patient into the best-in-class autologous bone substitute. And we'll talk a lot about why that is. We as a company, we sponsor a lot of research and studies. We have over 60 publications utilizing our protocol. We partner with tier 1 practitioners, key opinion leaders, and we are doing a lot of work in order to produce evidence-based outcomes and protocols that you can rely on. So this graphic is trying to depict a kind of an optimal way to evaluate and regenerate a defected site. So going from left to right, this is the site. This is, let's say, assuming this is a bone loss defect, which we are trying to regenerate and bring back to its normal dimensions and function. So typically we would use a bone graft or a bone substitute to fill the space, to fill the defect. And what we are hoping that's going to happen, and obviously using the correct materials and the correct procedures and the correct protocols, we are hoping that within 4 to 6 months, this site is going to start to regenerate through osteoinduction, meaning we're going to instigate bone regeneration regardless of the state of health of the patient. Obviously, some patients are generating bone on their own. They're healthy, they're young, but in most cases, patients do not because of some sort of compromise, medication, diabetics, obese, smokers, old. But so this is why it's so important for us to use osteoinductive materials that we know instigate bone regeneration. A lot of the graft is still going to be there after 4 to 6 months, and that's a good thing because that provides support to all that bone we just regenerate. Let's call it new formed bone, NFB. And that bone needs that support. It needs that support to mature, to go from woven bone, amorphic bone, to mature lamellar bone. And it's only that lamellar bone stage that now that bone can become self-sufficient. Okay, so that graft is needed to still be there to support that bone through that maturation process. And then 6 months after that, so we're now talking about a year, we are still going to see a little bit of graft, but we are now expecting that graft to start to resorb because now we've got the new form bone maturing, lamellar bone. It can start to kind of maintain itself, but now we just don't want to see the graft anymore. And so in about 10 months to 14 months, we want to see what you're seeing here on the left-hand side, right here, which is a defected site, fully regenerated, brought back to its original dimensions and function and aesthetics, and it's all bone, it's all native bone. And some of the things we're going to be looking at as we go through this process of evaluating grafts is inflammation, how much inflammation they're causing. We're going to look at how is the body reacting to it. Is it really considering it as a foreign body or not? The time it takes to regenerate, how long the bone is maintained, are we seeing secondary resorptions, and so forth. So if we look at the history of bone grafts a little bit for a second, people have used autologous bone that they harvested for a very long time, and it's a really good, it's a gold standard, it works really well. However, you need to harvest that bone. That means morbidity, that means a secondary site that we harvest from. And so it's not simple, it's expensive, it's painful for the patient very often, and it just takes time. So it's not very convenient, not for the dentist, not for the patient. And so industry came up with alternatives, and that is xenografts and allografts that I refer to as bone in a bottle, sourced from either animal bone or cadaver bone. But that bone gets processed the heck out. It gets really processed heavily through heat and induction and inoculation to make sure that there's no cross contamination. So at the end of this process, what you get is dead bone. And that bone, for example, xenografts just don't resorb. They're great as a scaffold, but they don't resorb. They'll always remain there. They go through a process of um, anyways, and the allografts are somewhat unpredictable, and again, some generate a little bit of bone, some don't. It really depends heavily on the patient. Another thing, the industry introduces synthetic fillers, and there's quite a few types of those. And again, um, we're not seeing high predictability using synthetics. They either resorb way too fast or they don't resorb at all, and we're not seeing them really generating the bone and the type of bone we are interested in. And so what Cometa Bio did is really we introduced a process to make autologous bone simple to harvest, and that's using the tooth. So really our protocol, which you'll see in a second, is taking the tooth that's extracted from the patient and converting that tooth in a very risk-free, clean way into autologous graft, dentin graft. Um, so ultimately, a bone substitute should really do four things. They should induce bone formation, and unfortunately, very, very few grafts on the market can claim that, if any. Okay, it's really just the autologous bone graft that can claim induction of bone formation, which is so vital, so important, especially if you're dealing with patients that are compromised. Then you want your graft or bone substitute to provide a good scaffold, not just for the immediate filling of the site, but also for the long term. And as I said before, to make sure that your woven bone, your premature bone that you are regenerating, if it's by natural means or by means of an osteoconductive graft, really take that bone all the way through to lamellar bone, mature bone that can self-sustain itself. And then the last thing I'll highlight here is that you at the end of this whole process, you don't want to see the bone substitute there anymore. You want that bone substitute to convert into native bone, and that's the process of turnover. So the graft kind of turns over, meaning resorbs completely. And again, some grafts do that, and some don't. Then a lot of people are saying, but wait a minute, you're using tooth for bone graft, and can you? And yeah, definitely you can, because when you look at the makeup of a tooth and you compare it to makeup of a bone, of an autologous bone, you can see the numbers. It's very, very similar. You get HA, you've got collagen fiber type 1, you've got some water, and you've got BMPs and growth factors. In fact, you've got more BMPs and growth factors per square unit in a tooth than you do in bone. So you have everything you need, everything is fresh, everything is clean and ready to go. If we are comparing bone substitutes or the main families of bone substitutes, and we're comparing it against this four criteria for what we're looking for in bone grafts, then we're seeing that the xenografts, although they are a great filler and a great scaffold because they just are there, it's good, but then again, they don't resorb and they don't really induce bone. So they don't induce bone and they never resorb. So we end up with with a filler that's going to be there forever. That's not ideal, not by any stretch of imagination. The allografts, there are a couple different types of allografts, but in general, they're not a bad scaffold. They're okay. They'll do some of the job. They're not going to really induce bone formation much, maybe a little bit. It's mostly on the patient, on how healthy the patient is. And then they resorb too fast. They don't sustain that bone for the long term. So that's not really good. Synthetics are highly unpredictable. And again, some are just resorb way too fast and some don't resorb at all. Some are good scaffolds, some are not so great scaffolds. Autologous bone is good. It's a gold standard, yeah. But the problem is when you're harvesting bone, you don't really always know the consistency of that bone. Sometimes it's more cancellous, sometimes it's more cortical. It's very hard to control. So, it does induce bone regeneration to a certain extent. But as far as resorbing, you never really, you can't really guarantee how it's going to operate. And it's also introducing a secondary site from where you're harvesting that bone and so forth. Autologous dentin, no secondary site, no morbidity, because it's a tooth that's coming out anyways. It's an excellent scaffold because hard, it's a hard substance. It's HA, dentin, the enamel. Of course, you keep the enamel. It induces a lot of bone, and all the research shows that it's really inducing a lot of bone formation. And because it's resorbing very slowly, but it does turn over, it is taking all that bone regenerated from woven bone to lamellar bone, and this is why our long-term results are so predictable and just consistently show almost no vertical loss and no horizontal loss whatsoever. So when we look at the studies that show everything I just talked about, those studies go back to the 1970s, but then Marshall Ewers introduced in 1998 a great study that talks about how dentin root matrix is a carrier for BMPs. So BMPs exist in dentin root, and that's one of the reasons why we're seeing so much bone formation as a result of utilizing dentin autograft. Then Schmidt in 2005 showed the same thing with growth factors, not just existing in the tooth, but also conserved in the matrix over time. So they are not degenerating, they're not reducing in time. And that's a really good thing. And on top of that, the dentin obviously is also ankylosis to bone, which is helping us with side dimensioning as well. And then since then, a lot of papers about the different protocols and different indications where they're testing and comparing, and they're doing split mouth studies between dentin and xenograft, dentin and allograft, and just seeing consistent results over and over again. So highly consistent conclusions every time with every paper. And the bottom line is dentin graft should be considered as equivalent to autologous bone with osteoinductive and osteoconductive properties. Okay, so let's look at the process. So the process takes 8 minutes. It's very simple. Most of it will be done by your assistant. And so from the time you extract the tooth until the graft shows up chair side on your tray, it should be 8 minutes. Okay, no more than 8 minutes. Okay, and this is the process. So once you take the tooth out, you extract, you'll take a high-speed burr. This is probably a step you will do, and irrigate it, and you're just going to remove any composites from the tooth. So amalgam and fillings and composites got to go. You're going to remove ligaments as much as you can. You don't remove the crown. You leave the crown. As far as decay, you do your best, but it doesn't have to be perfect at all, because this is not where we're cleaning the bacteria out. That's going to happen much later, and we have a really foolproof process for that too. So then you're going to dry the tooth, just because you don't want the particles to stick to the side of the chamber. You'll see that in a minute. You really want to get as much graft out of this as possible, so you want to dry the tooth real quick. And then you're going to take that unit, the base unit, which is this one, it's a smart dentin grinder, and you're going to bolt on top of it the grinding chamber. This is a chamber which comes sterile. It's one per patient. You can use it for as many teeth as you want, but it's got to be only for that patient. Okay, this is how we're maintaining the cleanliness and the autologous aspect of this. You don't want to introduce graft particles from one patient to another. This is why it's so important that you only use this for one patient. It looks expensive, but it's not. You're going to place the tooth inside that chamber, the grinding chamber. That unit is going to work for about 15 seconds, and during that 15 seconds, it will completely pulverize the tooth, and it will sort it into two specific particle size range. The top drawer here, the top tray, will give you the large particles, and the bottom tray you see underneath here will give you small particles. You then transfer those particles or graft into a mixing dish that's provided with the kit. You can see it here in the background. And you're going to apply this liquid, dentin cleanser, um, um, and you're going to completely cover the particles for 5 minutes. Then during those 5 minutes, this liquid will render the graft 100% bacteria free. So any decay, any any bacteria, any infection, the nerve tissue, any ligaments, any soft tissue remaining, all of that will be gone in during those 5 minutes. This is a high pH solution, sodium hydroxide and ethanol. You will then dehydrate that with a gauze. By the way, all I'm talking about here is done by the assistant, and typically they love doing it. And then you will then cover the graft, this time with this solution here, the green bottle. This is PBS, phosphate buffered saline. So this is saline which is buffered. And what this will do, it will wash out the dentin cleanser completely, and it will buffer the graft to pH of 7.2 biological pH. Okay, so at that time, this is what you're going to get in the dish. Graft that looks exactly like that. It's going to be nice, white, clean, bacteria-free graft that you can introduce immediately to the same patient or save it for later. So for example, if you're not using the whole thing, you can save whatever's left over for that patient for when they come back in 3 months, 6 months, a year, or in essence their lifetime. And we have a protocol how you keep, maintain, and sterilize the remaining graft. Okay. So what are the indications? Any grafting indication is this good indication for dentin grafting. I'll just mention a few that I like, especially like socket preservation. So the tooth is coming out and coming back to the same site, the same socket, to preserve and build up the bone. I say don't leave those untreated. Um, Attempt to graft those extraction sites every chance you can, because you're going to provide the patient with options for future. Even if he's not getting implants today or in the next 6 months, you're giving him bone and sufficient bone to be able to receive implants in the future. Full arch cases are wonderful because you're typically taking out teeth, and you typically need a lot of graft, and you get a ton of graft from extracted teeth. You get about anywhere between 1 to 2.5 cc's per tooth. Okay, 2.5 cc's up to 3 cc's obviously is a third or a second molar. Vertical and horizontal defects, those are best. That's where we shine. You're going to get extremely little resorption on the vertical aspect and no resorption on the horizontal aspect. It really works great and provides you with site stability right away. Perio defects is another one that I like a lot, where we see absolutely fantastic results, and obviously with dental implants in many different ways. So the main benefits of autologous dentin, just to recap everything I talked about, is the first and foremost the predictability, real predictable bone regeneration, long-term site dimensional stability. So the site is going to look the same as you first planned it, first grafted it, even after 5, 7 years and so forth. It's low cost because you're getting a lot of graft from a single grinding chamber. So, your total cost of grafting will be $50. No secondary resorption, so we're not seeing secondary resorption of the bone or the site after 6 months or 9 months or a year. Patients really like it because a lot of patients now care about where the materials that they're being treated with come from and what the origin is, and is it green, is it healthy, and they love the story. It's also going to help you sell socket preservation cases. You see a real accelerated site healing. You go straight to healing versus inflammatory process. Sometimes you don't even require barrier membrane. For example, if you can reach primary closure, you don't need membrane because soft tissue doesn't invaginate into autologous graft. This works really well with compromised patients such as diabetics, medicated. You can re-enter the site much quicker than you used to. So typically 2 months to 3 months, you can re-enter for your dental implants. And so for just a lot of benefits. And even I would say in many aspects, it's better than autologous bone because you don't need to harvest and you get the consistency, which you often don't get with autologous bone. So here's a case. This is done by Dr. Sifuni, Boston University, quite a few years ago. This tooth is coming out. It's highly mobile. It's exposed, and he knows he's going to have to graft this area here. Otherwise, this tooth is going to, he's going to have to extract this tooth very, very soon too, just because there's no support. So what's he doing? He takes out the molar, cleans it way too nice. I mean, this is way too much. He also decoronates it, which he was not supposed to do. Why? That makes no sense. I think he wasn't sure about the enamel, so he decided to decoronate. Here's the graft that he got. He's placing the graft in socket. He's using a membrane. I believe it's probably a PTFE membrane, and he's getting primary closure. So he doesn't really need the membrane either. He's way overdoing it. Okay, no need for a membrane because he got primary closure. This is the clinical view after 2 weeks. Look at that pink. Look at the healing. It's just incredible. And that's what we see every single time. And here you can see the before and the 3 year after X-ray. Look at the quality of that bone that he was able to regenerate here, and look how beautifully it reaches exactly where he intended it to be. And this is 3 years after, and that's very consistent. Here's another case, real quick. I'm just going to go through this quickly. These two teeth are mobile, chronic periodontitis. The patients wants it out. This is a 56-year-old male. So the doctor here decides to extract these two teeth. He's just going to use one of them, the third, to clean up. Here you can see it here as he's cleaning it up, grind and graft that site. He's going to place 2 implants here after 2 months, which you can see here in beautiful bone. And this is a 2-year follow-up. Speaks for itself. Just incredible bone quality. And that's consistent, case after case after case. So just to sum everything up, hopefully you got a good overview about different bone grafts and what they do, what they're supposed to do, what they should be doing based on your expectations, especially in more advanced indications. Hopefully you got a chance to kind of see more about our protocol and our device, the Smart Dentin Grinder, and how that works and how easy it is. So we really encourage you to consider it, consider integrating it to your practice. And we think it's a must-have product in your practice if you're extracting teeth and if you're replacing implants for sure. But even if you're just extracting teeth, so important to graft those sockets. So there you go. And it's an add-on to what you can offer patients. Patients love it. But if you're placing implants or if you're doing full arches, if you're doing on-4, on-X, if you're treating perio defects, it's really, I would encourage you to read the literature and see what this modality can really do for you. Okay, so give us a call or write to us, and we can help you get set up. Okay, buy your starter kit today. In any case, I'm going to see this is my last slide, and I'd like to open it up for Q&A at this point.